All right, so the last video we went over the liver's role in glucose, protein, and lipid metabolism. And here I want to look at this concept of liver detoxification. Um, uh, Ignore the notes here. It says phytochemical. We usually say most chemicals, and that could include things you take exogenously, like herbal chemicals, phytochemicals, or drugs or toxins and whatnot, things in your foods, as well as internal things like hormones, especially steroid hormones and metabolic wastes. All of these have to be metabolized by the liver and to some degree other tissues, and then excreted into urine and the bile. And there are two major types of so-called liver detox reactions, phase 1 and phase 2. And this is really another aspect of liver anabolic activity. We can think of when these detox pathways fail or are sluggish, we can think of this as being related to the Chinese concept of liver qi stagnation. So if you look at a substance like a exogenous toxin, phytochemical drug, or endogenous hormone, I should put that in here, comes into the liver. Now, this first part, phase 1, actually requires the presence of enzyme called CYP450s. It's also known as the microsomal mixed function oxidase system. These enzymes are concentrated primarily in the liver and the GI tract. So this happens not just in the liver, but in the whole GI tract. And what this is attempting to do is it attaches a polar functional group like a hydroxyl or an amine group directly to the lipophilic molecule. And that forms what's called an intermediate. So the intermediate's a little bit more water-soluble than the starting substance. But here's the problem. A lot of intermediates are actually more toxic than the starting substance. This is true, for example, of compounds in cigarette smoke and a lot of other, especially the cigarettes with additives in it, as well as many, many other drugs. And I'll look at Tylenol in particular. But the intermediates in the liver, this second phase can only happen in the liver, they have to be made more water-soluble and less toxic immediately by something known as a conjugation reaction, where you're adding a chemical group onto the original molecule. And this happens through, there are 6 different conjugation steps. Glucuronidation, sulfation, methylation, acetylation, amino acid conjugation, and glutathione conjugation. So for example, in sulfation, you're adding an SO4 group to whatever it is, forming a sulfated conjugate. And methylation, a CH3 group. This is a methyl group, so methylated conjugate. Glucuronidation, this is glucuronic acid, a sugar. Remember, this is how bilirubin is essentially brought from the immediate form that's not so water-soluble into conjugated bilirubin, is by glucuronic acid. Acetyl group is a little 2-carbon group. Amino acid, usually this is with glycine. And then glutathione is a larger antioxidant. So these are the 6 detox pathways. And you can actually, from some companies, get tests to actually measure your ability to use these 6 different pathways. And we can talk about the concept of a pathological detoxifier. That's a person with problems in one or more of these pathways. Where it gets complicated is that different substances like estrogens, remember there's 3 metabolites of those, and 3 main forms within many, many different metabolites, will be detoxed through different routes in different people at different rates. So this is where we all have unique biochemical profiles, and one of the big aspects of that is how well we get rid of and conjugate these molecules. Once they're conjugated, we can eliminate the metabolites through the urine or the bile. So here's a sort of overview, phase 1, intermediates, phase 2, conjugation, and then eliminate through the bile or the urine. I mentioned that in some cases, the intermediates can be more toxic than the original starting substance, and that's true for paracetamol or acetaminophen, same thing. This is Tylenol. So one fate of Tylenol is that it can go through the CYP450s, and this is called N-hydroxylation, and it results in this molecule called NAPQI. And it turns out NAPQI is horribly toxic to liver proteins and the nucleic acids, so it destroys liver cells. So in order to make sure this doesn't happen, a person needs to have adequate conjugation pathways, in particular glutathione conjugation. So you can conjugate NAPQI with glutathione, and this is non-toxic. Paracetamol, Tylenol, can be actually glucuronidated and sulfated as well, but these are usually very slow pathways in most people. So what can happen in some people when they take too much Tylenol is that, or if they have slow glutathione conjugation, you can build up NAPQI, and that can destroy the liver, and sometimes within hours. People can go into complete liver failure within hours of taking the Tylenol. So this is a huge thing that people need to be concerned about. Now, in the hospital, interestingly, what they will often do is give you, often intravenously, a precursor to glutathione, which is N-acetylcysteine, NAC. So NAC is one of the building blocks of glutathione, and so this can be administered intravenously. I often have patients just taking it because it will help to speed up the conjugation pathways. This is a very, very complex area in terms of, I don't think I have the notes on it. Yeah, I don't have it really here. Very complex area in terms of liver detox because there are many nutrients which can upregulate or downregulate these different pathways, as well as different herbal molecules which can upregulate and downregulate. So this can be both beneficial, can help you detox things faster, but also if a patient's on medications and you start messing with these pathways, you can actually, in some cases, have them metabolize the meds faster, so you really don't have any effective medication in the system, or inhibit their metabolism and they build up the toxic levels. So one interesting example is St. John's wort, Hypericum, and birth control pills. St. John's wort will increase the activity of the phase 1 enzymes, and you will process your oral contraceptives faster, and they'll become ineffective. And so this is something to warn people about. We really don't know how all the different Chinese herbs in the dosages that you typically use affect CYP450 enzymes. So that's where it gets very complicated. But with most drugs, you can actually look it up in the database, know exactly which CYP450s they affect, and how that might influence other medications. I'll just say here, before moving on, is that there are 6 isotypes, 6 isozymes of the CYP450. So there's 6 major ones. For example, CYP3A4 is a major one, and so on and so forth. And there's 6 of these. And those 6 are the major ones we're concerned about with drug metabolism. I should say in general, there's well over 100 different isozymes of CYPs, but for drug metabolism, we're concerned with 6. So someday we might have lists of Chinese herbs and the 6 isozymes and be able to predict all that, but currently we do not. Okay, so I'll just maybe go on a little bit here and talk a bit about the bile as well. Mention a lot of these substances have to be put into the bile for elimination, but I want to look at the composition here of the bile. So the bile is really synthesized in the hepatocytes, and those specialized hepatocytes called cholangiocytes, and they enter the bile ducts. And the bile acids really comprise about 50% of the bile. So half the bile is bile acids. And the bile acids are made from cholesterol. And there's 2 major ones, the cholic and chenodeoxycholic acids. These are synthesized from cholesterol. When they are bound to cations like sodium, they become bile salts. So the difference between a bile acid and bile salts is that the bile acid has this carboxylic acid group, and the hydrogen dissociates, and so you get a negative charge on here. And if you have a salt like sodium in here, these will form an ionic bond, and you'll form a salt. So the bile salts and bile acids are very similar this way. Gut bacteria further dehydroxylate the cholic and chenodeoxycholic acids into deoxycholic and lithocholic acids. And then in the liver, you get further conjugation into all these conjugated bile acids. So it gets kind of complicated, but there's a lot of different bile acids here and that are then going to be put into the bile. And their role, remember, is essentially to act as amphipathic molecules to emulsify lipids. So these are going to get squirted into the small intestine, and then we already went through this where the lipid droplets then will be surrounded by the bile acids, and this will form a little micelle. And the micelles, then the lipases can go to work, start cleaving off fatty acids from that, and they can be absorbed. Okay, so that's bile is 50% bile acids. There's also bilirubin in bile, and I think I've gone over that numerous times. This is, of course, the breakdown product of heme from hemoglobin. And in the liver, it's conjugated with glucuronic acid to form conjugated bilirubin, so that's what you see in there. And this is a bile pigment, so that gives color. And this will get further metabolized to give your stool a dark color. Some of it gets reabsorbed, and it's eliminated through the urine, and that gives your urine the classic yellow color. Cholesterol is also found in bile, and this is important because this is a major route of elimination of cholesterol. So I said we make the majority of cholesterol in the liver, but we also eliminate it through the bile. So if you're having bile problems, sluggish bile, and that's something known as cholestasis. If you have cholestasis, cholesterol will essentially build up the levels that you don't want. Also, we'll look at this shortly, the cholesterol has to get eliminated into the small intestine and then move all the way through. Unfortunately, a lot of it gets reabsorbed, and so if there's not enough fiber in the diet, fiber actually binds all of the bile up. Not enough fiber in the diet, you can get too much cholesterol. That's why adding fiber to the diet can actually lower cholesterol. Lecithin is a phospholipid inside of the bile, and this keeps the cholesterol from precipitating out, so there's a sort of inverse relationship between these two. Electrolytes are also in the bile, like sodium and calcium, and then a lot of fat-soluble metabolites, a lot of those detox things I talked about. So for example, estrogen conjugates, testosterone conjugates, cortisol conjugates, et cetera, et cetera. I'll just mention again, many plant molecules, if you ingest the plant and then measure in the blood the presence of the different molecules from that plant, you'll find they actually are in the form of conjugates, not as the primary molecule, because the liver's already gone to work on it. There is a recirculation of bile, so about 95% of excreted bile is reabsorbed in the ileum and reused. And so we actually can recycle the bile salts. Only about 0.5 gram is lost in feces per day. The bile acids are carried by albumin back in the portal blood to the liver. Cholestyramine, which is a medication, and then fiber bind the bile acids, and that prevents cholesterol reabsorption. So this is why you can eat oatmeal, for example, to lower cholesterol. There's different factors influencing the release of bile. So that would be cholecystokinin. I've already talked about that. That's one of the strongest stimuli. Secretin. Remember, this is the one that increases also bicarbonate secretion. And then the parasympathetic nervous system stimulates gallbladder, smooth muscle contraction. So those three influence the release of bile. Bile is stored in the gallbladder until needed, and the maximum volume is about 20 to 60 milliliters. But you can actually have about 12 hours of secretion, or 450 milliliters stored in the gallbladder. Water electrolytes are reabsorbed, so the bile is really concentrated in the gallbladder. You can add water to it and then eliminate the bile. And that's why, unfortunately, if you concentrate the bile, especially if you have low lecithin, low phospholipid, cholesterol might precipitate out and you might get cholesterol stones. I'll talk about gallstones, but cholesterol stones are by far the majority. You can also get stones from bilirubin. Those are called pigment stones. Those are much more rare. So the functions of bile here, it's helping to get rid of extra cholesterol and fat-soluble wastes and toxins via the feces, as long as you have high fiber in the diet. It makes the cholesterol more soluble, so it prevents this precipitation of cholesterol in the gallbladder, and that's via the phospholipid. It also facilitates fat digestion, absorption, and that we talked about that with the forming of micelles. And it facilitates absorption of fat-soluble vitamins as well, so you need adequate bile function to get vitamin A, vitamin K, E, and D from the diet. This is just a review. You can read this. I've gone over this many times, of bilirubin. And so if you're unfamiliar with bilirubin, I would just recommend that you review this. Okay, so a couple of definitions with bilirubin. Hyperbilirubinemia refers to increased concentration of bilirubin in the blood. And this can actually lead to jaundice, where you have signs and symptoms of hyperbilirubinemia. Cholestasis, presence of bile plugs in the bile canaliculi and visible bile pigment in the hepatocytes. And then cholestatic jaundice is histological cholestasis and hyperbilirubinemia. So just a couple of definitions there. Other functions I didn't really mention the liver would be storage of your fat-soluble vitamins, iron, copper, and B12, and then I mentioned the formation of blood and the immune function. So that's an overview of all of the different liver disorders. And what I'm going to do for the purposes of this week actually is I'm going to actually go over all of the liver and gallbladder diseases next week. So we're going to stop the videos here, and then I will post next week the liver and gallbladder diseases. And you'll have one more quiz to do, and that will be over all the liver physiology and gallbladder physiology, as well as the pathologies, which will be posted next week. So stay tuned for that. So go ahead and just review all the notes I've gone over so far. You could study that. And then we'll finish up the liver section next week. And then I will post the notes for the kidneys, which you're welcome to read. But the kidney information, I won't have a lecture series on that just because we're running out of time at the term, but you can have that for your own, and we can talk a bit in class about those conditions. So stay tuned, and hope you enjoy the rest of the videos.